then I suggest we immediately get going with the educational starter. So those of you who have been in this seminar before know that well that we also always try to, depending on the people who present here in the seminar, give a bit more of a basic introductory lecture part where the technique that is used by one or several of the speakers is explained a bit more in detail. And this is what I'm trying to do now. And the educational starter is about isobaric tagging in proteomics. How does it work? Maybe some of you here in the audience have never heard about that, and that's why I want to explain it a bit more in detail. Okay, so if we do proteomics, or actually also in metabolomics, it's very similar. There are 3 different quantification units that you can derive or use from the LC-MS data that we acquire. You all know we use the data to identify peptides or metabolites, but we also usually always want to quantify what's inside. And there are 3 different, conceptually different units you can exploit. I would say still the majority of papers that get published use as the quantification units from our LC-MS data chromatographic peaks. And this is also what Flo just showed before in his talk. He also always integrated. So what you do here is you use the fact that we always add an LC system in front of our mass spectrometers, and you follow how the analytes of interest, whether it's peptides or proteins, dilute over time from this LC system, and then you can take the peak area or the peak height and use that for quantification. That's used a lot, and that's working great. However, there are also other units you can exploit, and another unit that proteomics people also acquire is spectrum counting. So we acquire these fragment ion spectra of all our peptides. And what you can do to quantify a protein is you can simply count how many spectra in an LC-MS run do identify a certain peptide from a certain protein. And protein A here has 10 spectra, protein B 7, protein C 3, and then you can use this number as a quantification unit. The cool thing about spectrum counting, it's super easy. You can do this very easily with the data. You don't need any great algorithm. For chromatographic peak areas, you need an algorithm that explains the start of the peak, the end of the peak, and does the integration. It's computationally much more difficult. So spectrum counting is very easy. However, you can imagine when you do not acquire a very large number of spectra, spectrum counting very quickly is limited in its accuracy. So if you just acquire a single spectra for a protein, that's not a very good quantification level. So in reality, I think spectrum counting is rarely used in the proteomics field. There are some specific applications that still rely on it, but yeah, people usually favor chromatography-based quantification, or they favor what is called isobaric. And this is now the 3rd quantification unit that we will talk in the next slide about. So what's isobaric tagging? And this is also, by the way, what looks. The isobaric tag, exactly. That's what Flo uses, what you will hear about in the second talk. So where does the name come from? Isobaric tagging. Maybe you're familiar with the term isobar from the weather report. So in meteorology, people talk about isobars when they talk about lines on the map of equal or constant pressure. So something is equal over a certain area. And that's the analogy where people call isobaric tagging because in isobaric tagging, we work with labeling reagents that all have the identical total mass. So you work with different reagents that all have in common that their total mass is constant, is exactly the same, just as having the same pressure on a graph. However, each of these reagents that we use have a different distribution of heavy atoms within the chemical structure, and we will look at that in detail. So every isobaric labeling reagent is a three-functional reagent consisting of a reporter part, a balancer part, and a reactive group. And the reactive group that people like to work with is an amine-specific reactive group that covalently binds to primary amines. And in proteomics and in bottom-up proteomics, where we generate peptides, we always have primary amines at the N-terminus of all our peptides, as well as at the C-terminal lysine residue. The side chain is also a primary amine. So if we treat peptides with such a reagent, every peptide will be labeled at least once at the N-terminus, And all the peptides that carry a lysine residue will also be labeled as the C-terminus, so will be labeled twice. And the only thing that's now really important to understand about isobaric labeling is that we use different reagents for one experiment, and the sum of the masses in these reagents is always constant, okay? However, the individual masses of reporter and balancer are different. There are two commercially available substances you can buy on the market. One is called TMT reagent, the other iTrack reagent. You can see here their chemical structure. They are very related. We have the reporter group, balancer group, and this reactive amine reactive group. This TMT reagent is the most successful one on the market. The link make more different versions of it, and it's commercialized by Thermo, a company very popular in proteomics, and this is what most people work with. Also, Flo has used TMT labeling tags. So here you see how they look like. This is an example for a 6-plex TMT experiment, which means you will work with 6 different reagents, and each of these reagents now has different heavy atoms in its structure, and the heavy atoms are always indicated with such an asterisk. And what you can see is that in reagent number 1, in the reporter ion region here at this area, there is not a single heavy atom. So the mass of this reporter area is 126. And there are a lot of heavy atoms in the balancer region, so the mass is 103. With the second reagent, now 1 heavy atom moved into the reporter area, and we lost 1 heavy atom in the balancer, which makes a 1 M/Z difference between compared to the first reagent. And this goes on for all the 6 different reagents. We have always 1 more heavy atom in the reporter and always 1 heavy atom left in the balance. Important thing, the sum of reporter and balancer is always constant, but individually they are all different. Okay. If you got that, you got TMT. This is what it's all about. This is an example for 6-plex experiment. In our days, you can commercially buy 18-plex TMT. So you can buy up to 18 different reagents. What people have started doing is they have also started to differentiate between a heavy carbon and a heavy nitrogen. So you can isotopically resolve differences in heavy carbon and heavy atom. Heavy carbon and heavy nitrogen. However, to do that, the mass difference is very small, only 6.3 millidalton, and you need very highly accurate mass spectrometers to be able to measure 18-plex or TMT 11-plex is what we do a lot still in the facility, compared to TMT 6-plex, where you have always 1 M/Z difference. So in a way, you can understand TMT reagents like barcodes. You can barcode different samples with different reagents. So with TMT 6-plex, you could perform an experiment that would look like this. You would work with 6 different samples, can be 6 different proteomes, can be 6 different cancer cell lines, can be 1 cell line treated with 6 different doses of a drug. Then you would extract the protein from each sample, digest each protein per proteome with trypsin, and then you would react each of these 6 different samples with a different TMT labeling reagent, okay? Which will mean that the peptides in these 6 different tubes will all be labeled now completely at the end. Those would have a C-terminal residue also at the C-terminus with its individual reagent. Then you can put all the 6 samples together. And then from that moment, when you have them together, you can perform the rest of the workflow in 1 sample. This is also what Flo just talked about, stable isotope labeling method. Once you have the analytes of interest together in 1 sample, everything that happens to the sample from that moment on will be constant and will be accounted for as they are all treated in 1 go. So you can do peptide fractionations on these samples. You can do phospho enrichment. This is something that Flo has done and will show you in his talk. And you will be able to measure very precisely then the quantity of these peptides in the sample. And what happens now if you take such a pooled sample and shoot it into the mass spectrometer? Well, what we will see is that if we measure the MS1 full scan, which shows us the complete peptides, each peptide of each of these samples still has the same mass on MS1 level, because here we have always the same total mass of each reagent. So for each signal that we will see in MS1 scan, we will actually see a compiled intensity for every peptide by summing up the intensity of every individual cell. Does it make sense? Because the mass is still not different between them. However, if we now isolate our peptides and fragment them in the mass spectrometer, these labeling reagents have been designed in a way that upon fragmentation, the bond between reporter and balancer will break. So this bond here will break, and we will get a free reporter ion produced. And this free reporter ion we will see here at the very low mass range of our MS2 scans. And these reporter ions now we can use for quantification. We can differentiate a peptide coming from first, second, the third, or the sixth step. While all the other ions that are also produced are used to identify the underlying peptide, the identity of the peptide, as we always do that in proteomics. Okay, so summary, what are the pros of isobaric labeling? Well, the great advantage of isobaric labeling is the multiplexing. We can take many samples, up to 18 routinely these days, put them together in one sample, and therefore increase throughput of your sample preparation and your measurements. You reduce the analysis time, unless you do very extensive peptide fractionation and take one sample and again put it into many different fragments. Then this reduced analysis time is maybe not so great. You have smaller run-to-run variation because you have good quantitative precision, and you have less missing values in your data sets when you use TMT labeling. And it's applicable to a wide variety of samples because you add these reagents on peptide level after the peptides have been extracted and digested. So you can do TMT labeling also with cultured cells, with human tissue, with plasma, with any sample type. However, there are also downsides. Isobaric labeling is rather expensive. So the official amount you have to pay when you buy a TMT labeling kit is in the range of 500 to 1,000 euros to do one of these experiments. And you have a limited quantitative accuracy in TMT labeling because of an effect that is called ratio distortion. And this I try to explain here. So one problem that we have in TMT is that every peptide is labeled with the same tag, right? So if we put the reagent, our TMT reagent in one sample, every peptide will carry the same reporter ion, the 126, for example. And what we do inside our mass spectrometer, if we isolate a certain peptide of interest to do then an MS2 analysis on it, if we have very complex samples, we are unfortunately not very good in isolating just a single peptide, but maybe we have a contaminant in there, which will also carry the exact same tag. So the red guy here should be a small contaminant, the black guy is our peptide of interest. And if we then look at the formed reporter ions upon fragmentation, you see that this red guy is always contributing to a certain intensity there. So the ratios that we will measure between our peaks is actually not 100% accurate. And if it's true, so what tends to be the case is that interfering peaks are often maybe constant over a lot of samples, not necessarily always, but a lot of proteins usually are constant in samples. And usually if we have a constant peptide, a constant interference, and a regulated precursor, the ratios that we will measure with our reporter ions always tend to be too small. So we often have ratio compression. However, if we are lucky and have a peptide that is a contaminated peptide that is very highly regulated, we can also have a ratio expansion. But sometimes this is why it's called ratio distortion. The wrong ratio tends to be rather compressed most of the time. And that's a problem because, we are not as accurate as we would like to be. And also the quantification range, so the lowest and the highest abundant protein or peptide that we can measure is limited in TMT. So people have thought a lot about this disadvantage and tried to improve that. And one way to improve the accuracy in the measurements with TMT labeling is to do MS3 measurements. So what that means is we do measure an MS1 scan on our sample, then we use the collision energy that does not cleave of our reporter ions, so that fragments only our peptides, and we get all the different peptide fragments. And then we just focus on one of these fragment ions and fragment this fragment ion again in an MS3 scan. And only at that point we cleave off the reporter ions. And why is this advantageous? Because there is actually, it's less likely to still have interferences here on an MS2 level than on the MS1 level. So the ratio compression, ratio distortion issue tends to be lower with such an MS3 measurement. So you have reduced unwanted co-isolation of peptides, a better accuracy, but you very much lose ions on the way from MS1 to MS2 to MS3. So only 5 or less than 5% of the signal intensity really reaches the detector at the end of the measurement queue. And doing this series of measurements is slow, so you tend to identify much less peptides in the MS3 mode than you would if you would not do this additional step. And then there is my last slide here, where I just want to mention too, because this is also the way how Flo has done his measurements. So this problem that you lose a lot of ions from MS1 to MS3 can also be improved if you work with triplet mass spectrometers by doing something that's called multi-notch MS3. And what you do here is when you select a certain peptide in MS1, you have on that instrument the option to also select not just one fragment ion that is formed, but several. So you can up to 10, or 10 is what we use in most of our methods. You can select 10 different fragment ions, and then the intensity in the MS3 mode will be then again the sum of the 3. And like this, you again manage to get more ions to the detector and reduce a little bit this problem of sensitivity. Exactly, increased sensitivity, but multi-notch synchronous precursor selection can only be carried out on these specific mass spectrometers that allow these measurement modes. And these are these orbitrate triplet mass specs, which are very expensive, but luckily we have one here also at the core facility. Okay, yeah, that's all I wanted to say about
Florian Bayer. So I'm really happy that we still got you as speaker. We already tried him as a last-minute speaker when we had a sick person, but that unfortunately did not work, but we still got you. Happy to have you here today. So Florian studied food chemistry here at TUM, did his bachelor's and master's, and then joined the group of Professor Hochmann, defended his PhD this spring, in February, and now he is about to go out to industry or consulting, switching gears a little bit. So I'm curious to hear what the future will bring you. But today I'm really curious to see what you've done with your postdoc project at the chair.
Basically, it was a lot of PhD.
Okay, sorry.
No worries. Yeah, thank you very much for the kind introduction. Also from my side, welcome to more or less my final show, as I will leave BayBioMS very soon. And today I'm very happy to present you some parts of my PhD topic, especially how we could decode the aroma of pea proteins and use this knowledge for further flavor optimization.
You have to pick one and choose a presentation.
So first of all, I want to start with some introductory words. I think all of you know that we are really facing a lot of different challenges within the next years. So with a growing world population, there are predictions that till 2050 we will be 10 billion people on this earth. Moreover, there is for sure the climate change. So 25% of the greenhouse gases belong or arise from the food sector. Then we have a lot of food loss. Almost 1/3 of our food is wasted, even though or despite there is a high quality and it's still good. And last but not least, there are a lot of nutrition-related diseases. So due to a not balanced or wrong diet, a lot of people are suffering adiposity, obesity, CVD, so cardiovascular diseases or diabetes. And yeah, 2 billion people are overweight, facing 800 million undernourished people. So there is, or we are in the situation that we should act. So to start, but also to start with some good news, is that there is already a quite high consumer awareness towards alternative proteins. So as you can see on this slide, the BCG Blue Horizon study from 2022 highlights that a lot of consumers already know alternative proteins or food with alternative proteins as ingredients. So worldwide, around about 76% are familiar with these food categories. But one main requirement for a successful alternative food or food product is that they show a good and convincing flavor. And then there is a pretty high potential of around about plus 100%. But when we are talking about flavor, what do we actually mean? So flavor is not only the aroma part, neither only the taste part, nor the trigeminal chemosensation. It is all of them, which makes it very complex and difficult to capture the overall flavor of a food. So we have the aroma part, and in our body we have about 400 different odorant receptors, and for sure one main requirement for a volatile is the volatility. So if the odorants are not volatile, they won't get into our nose and to the odorant receptors. Besides, we have the 5 classical taste attributes and senses, so bitter, umami, sweet, sour, and salty perception. And to make it even a bit more complicated, we have the so-called trigeminal chemosensations, or the organoleptic sensation, which arises by the perception of the nervus trigeminus, which is located round about here. And with that, we can perceive sensations like spiciness induced by capsaicin, chili, spicy taste, pungent taste, for example, by mustard, but also cooling effects by menthol, for example, when you are chewing a chewing gum. So flavor is a highly complex interaction between aroma, taste, and the trigeminal chemosensation. And as first experiment regarding these alternative proteins and the P proteins, we started with comparative flavor profile analysis. And the results you can see in these bar charts. So we got a lot of different commercially available P proteins around Europe, and did this comparative flavor profile analysis. And you can see that on the left side the taste, and on the right side the aroma part, all of them show very similar attributes, but nevertheless there are some distinct differences. So especially regarding taste, P proteins are characterized by a slight bitter astringent off taste, and regarding aroma, there are a lot of grassy, beans-like, honey-like, nutty, malty, and earthy aroma notes. So all of these commercially available P proteins are currently not that ideal to use them as food ingredient, especially in completely different categories, because they will always introduce kind of nut flavor. So for that, we wanted to decode this off aroma of P protein. I will focus on the off aroma because a former colleague, Peter Glaser, focused on the off taste part, but I will not get deeper into detail regarding taste. So with the help of the Sensomics concept, we could decode the aroma of P protein, and the Sensomics concept normally starts with a separation of volatiles from non-volatiles, a structure identification, as well as structure determination. But as especially P-related matrices and P proteins are pretty well investigated regarding the qualities or regarding aroma compounds, we could really jump into the targeted analysis of these described analytes. And with these, or we developed a quantitation method based on stable isotope dilution analysis. And with the help of this method, we could quantify all of the corresponding odorants. In a further step, we then introduced the order thresholds and calculated so-called dose over thresholds, so DOT factors. And per definition, a DOT factor bigger than 1 means a direct contribution to the overall flavor or aroma of a product or food. So because then the concentration of the odorant is exceeding our human threshold and we will perceive it. And based on these DOT calculations, we can then do recombination experiments by sensory evaluation, but as well as omission experiments. And you will also see some Sensomics mapping within this presentation of the technological process. So to start with an overview, on this slide, you see different aroma-active compounds described in literature for P-related matrices or P proteins. And all of them were quantifiable with the help of a newly developed 3NPH LCMS method, but also we also used a dairy LCMS method to capture some others. On this slide, you can see the sample preparation of the method, so of the stable isotope dilution analysis, and we started within a first step to suspend the P proteins in acetonitrile waters and the solvent and added our internal standard mixture, so an isotopically labeled internal standard mixture. With that, we could, after homogenization and equilibration, we could get a constant ratio between the analyte, which is present naturally in the P protein, so for example, the diacetyl here, and the isotopologue, so diacetyl with strict deuterium effect. With this constant ratio, all further analyte loss could have been fully compensated, making the stable isotope dilution analysis a very precise method. And after derivatization, which you can see in this blue box here, we could make these volatiles to non-volatiles with the help of 3-MPH, this molecule. And these non-volatile compounds were able to be quantified with the help of liquid chromatography, which enabled us to speed up the analysis, especially compared to S chromatography analysis. On this slide, you can see different MIM transitions of the orderants as well as the internal standards used for quantitation. And all of the orderants have been validated regarding recovery rates, so in a low and high spiking experiments, as well as the limit of detection, the LODs and LOQs, so the limit of quantitation. And with the help of the stable isotope dilution analysis and internal calibrations, we could quantify several analytes, for example, here in the Neutralis S85F P protein, and introducing the order thresholds here, for example, in determined in water, we could generate out of the quantified concentrations these DOT factors, the dose over thresholds, leading us for Neutralis S85F to a list of 27 analytes with a DOT factor bigger than 1 and potentially impacting the overall flavor as a consequence. For example, 3-methylbutanol showed the highest DOT factor with more than 10,000, and as next task, we wanted to see if these 27 analytes are really enough to reproduce and re-engineer the authentic aroma of P protein. So we created an aroma recombinant based on these 27 analytes and compared it in a sensory evaluation in comparison to the Neutralis S85F P protein, which is presented in this darker green color. And you can see that there is quite a good fit between the complete recombinant in the slighter green and this darker green, so the reference. But as 27 analytes are pretty complex and it's really taking a long time to weigh all of them into the recombinant mixture, we wanted to know if you can further reduce this complexity and performed so-called omission experiments. So complete aroma recombinants have always been sensory validated or evaluated by means of triangle tests against incomplete ones. So for example, this green pie chart was omitted, and if the panelists recognized a significant difference with at least a p-value of 5%, we could assume that this green pie chart is a P-food orderant within this recombination and thus within the P aroma. In doing so, we could further reduce these 27 analytes to 9 KFOs, namely 3-methylbutanol and heptanol with very highly significant differences, 2-methylbutanol with a highly significance, and hexanol, acetaldehyde, PE2,4-nonadienol, E2-octanol, benzaldehyde, and nonanoic acid with at least significant difference. In the next step, we then wanted to know, are these 9 KFOs really enough to do kind of a minimal recombinant and still get a convincing aroma profile in comparison to the reference, so the Neutralis S85F protein, as well as to the complete recombinant. And this is represented with these dotted lines, and you can see that these 9 KFOs are really enough to re-engineer authentic P protein aroma, underlining once again their role as P-food orderants. But we performed all of these Sensomics aroma decoding just with one P protein, the Neutralis S85F P protein. And as you saw within the first slide, we had a lot of different commercially available P proteins. Therefore, we wanted to know how representative are these 9 KFOs in comparison to these other P proteins. And for sure, we couldn't do all of these sensories for all of the different P proteins, because then probably my time would here be a bit longer. Therefore, we wanted to see if we can see any trends based on the DOTs within a statistical cluster analysis of a heat map. This is basically shown on this slide, and you can see that especially the aldehydes, so this substance class, automatically clusters together within the left part of this heat map. And moreover, in this cluster, you can find 8 of 9 key food orderants identified in the Neutralis S85FP protein. Therefore, we assume that these 9 KFOs are general KFOs among different commercially available P protein products. And now I also want to tell you a bit of how we could use this knowledge for further flavor optimization. So another part of my PhD was in cooperation with the chair of Food and Bioprocess Engineering, to develop a fat-reduced milk dessert or dairy product with the help of pea protein as a fat replacement. So in comparison to a full-fat milk dessert with 50% of fat, we created one or several milk desserts with the help of pea proteins, lacking of the half of the fat. So in the end, there was a fat content of 7.5%. In the first step, comparative flavor profile analysis were performed. And so in this dark green color, you can see the reference, so the full fat milk dessert, and all of the other ones are fat-reduced milk desserts with the help of pea proteins. So taking a look at the left part, again, the taste part, you can see that an introduction of pea protein within this milk dessert system doesn't influence the taste attributes significantly. But for sure, taking a look at the aroma part, you will see that especially within this prototype, there is a very highly unwanted grassy green bean-like off aroma within the milk dessert. But with the help of optimized pea protein microparticulates generated by the chair of Food and Bioprocess Engineering, we could clearly reduce this off flavor, and the best one was within this blue-colored PMD, and showed almost no grassy green bean-like off aroma. And how could we achieve that? So with the help of this newly developed LCMS method, we could take a lot of different samples and screen almost every process step, starting from the pea protein educt over the extrusion, so the microparticulation, where the pea proteins were transformed to fat analogs, over the drying and the partial fat reduction. And with this, we could see that especially this drying of the microparticulates before introducing them as a fat replacer almost gets rid of the essential concentrations of these 9 KFOs. And therefore, we could clearly reduce this off flavor within our spray-dried microparticulated pea protein milk dessert. So to sum my presentation briefly up, with the help of the Sensomics concept, we could decode the aroma of pea protein. But Sensomics also helped for sure to decode other flavor, taste, and aroma of foods. But the novel aspect was that with the help of these unified flavor quantitation methods, we could really speed up the Sensomics decoding and therefore generate big data. And with this big data, we can, in the end, optimize breeding programs, select the right raw materials, optimize as well technological processes, in the end, optimize the flavor of different foods. So I'm at the end of my presentation. Here you can see some former colleagues or still colleagues, at least for 2 days. And I also want to thank you for your kind attention. And
Florian Bayer. Flo, in his own words, is a complete inbred of BioM. So he also, just like the others, Flo, studied molecular biotechnology, bachelor and master. He did his master thesis in the group of Christina. That's where he learned proteomics, and we really liked it, and that's why he decided to stay for his PhD, which he started 4 years ago, roughly. And in the last 4 years, Flo was really impressively successful. So he's already the author of a series of great publications, but the top one is the one we will talk about today, the one that was published in Science, and which entails the major part of his PhD, right? So a large part of your PhD and that work. And yeah, the aim of this work is to figure out and to entangle how drugs act on living cells by doing time-resolved proteomics. Look, we are very happy you agreed to be here, and the stage is yours.
Yeah. So yeah, hi everyone. Also from my side. Just one thing. My voice is a little bit damaged. I hope it will survive the next 20 minutes. But yeah, so I hope at the end everyone will really like dose-resolved proteomics, which is very different than what most people do. And but it's really cool. You will see it in a second, and I will give you all the details. So, and we call this technology Decrypting. So decrypting PTMs and drug actions on the entire proteome. And before we really go into details, I just thought let's start a little bit smooth. So we need to click once, right? Like this. Ah, yeah, perfect. So before we go there, I just want that everyone thinks about his or her, how should I say, favorite protein. Yeah. So just think about going your cell and think about your favorite protein. How does it look? Yeah. Just imagine the 3D structure. And I bet if you do this, it looks something like this. So this is hopefully not what you thought about. It's a spike protein of COVID, but I think pretty sure you had something like this in your mind. The reality, however, is very different. This is how probably in reality your spike protein looks like. And what you see there in blue is it's heavily glycosylated. And so, in fact, most of the proteins that we have, they're actually heavily modified. And you should think about this because this is important. And glycosylation, obviously, is not the only modification that exists. So this is also not a complete list, but this just gives you an impression of the variety of different modifications that can happen on different side chains of proteins. And what is also true is that if we now look in the entire proteome, then every protein basically carries post-translational modification. So it's not a weird esoteric something like, no, this is like what's always there. And this is what really shapes the function of proteins. And this is also when we just go to this general view, why proteoforms are so important. And when we just look at the human gene, for instance, it has basically 20,000 genes. And that alone, I mean, people were really surprised when the Human Genome Project was around that it's just so few genes and humans are still so complex. But it's just a fact that we have then isoforms that maybe get you something like 100,000 different versions. And then you have the proteoforms, which I think is the coolest of all of this. Because you can get, I mean, people don't really know how many there are, but for sure it's more than 1 million different proteoforms that exist. So a proteoform is just basically the summary of all the different steps from one gene over an isoform over different PTMs. And then the other important thing is that when you look at these different proteoforms is that, yes, they're coming from the same gene, but it probably has a very different function. So if it's phosphorylated in a certain position versus it's maybe or in this case, it's a glycosylation, like glycosylated somewhere else, then they probably have a very different function or localization or interaction partners or lifetime or enzyme activity. So you have the entire zoo of things that can happen. And now that we agree on that protein modifications are really important and we should always look at PTMs, it turns out that when we now do mass spectrometry on them and we look at different PTMs, and it's really sad to know that actually just 1 to 5% of the things that we can see now with mass spectrometry is actually characterized. So there's really a need of looking at PTMs and how they are doing and how they are involved in different things. And on the same hand, we have human drugs. And many of the human drugs, they're for sure often interact with proteins. And in fact, if you, for instance, go for protein kinases, which I like the most or work the most, the job of a protein kinase is just changing one proteoform to a different proteoform. Yeah, so it's just taking the substrate, adds a phosphorylation, and then you have a different function. Usually it's inactivating or activating substrate proteins. And these drugs are actually well established in the clinics, but we really do not know how they exert effects on a global proteomic scale. And this is what really Decrypting is about, because we really want to understand this interplay between drugs and PTMs. And when we learn more about the PTMs, we learn more about the drugs and vice versa. And this brings me to the next slide, where we really need now some sort of concept or workflow that really enables us to dig deeper into this PTM biology. And what we are doing is we start with cell treatments, and I talk in a second about why we do this dose dependently. So we just treat our cells, then these proteoforms inside the cell, they change. And then we very quickly lyse the cells, we get our proteins, and there's a lot of optimization that happened over the last years. So this is kind of our current or most relevant workflow at the moment. So we do this SDS lysis that's like really, really harsh and gets you all the proteins, and it's better than this urea protocols that had before. And then combine it with the sp3 cleanup, because SDS is not really so compatible to mass spectrometry. And then we obviously do bottom-up proteomics. So we need trypsin to get our peptides, and then we get a slew of peptides that have these modifications. And now comes what Tina so nicely explained with the TMT labeling. So we take our peptides, each different dose that we had will get then a different bar that we will read out later. And there's lots of reasons why I think phosphoproteomics and PTMs are really a nice match for TMT, because you can really boost your starting material. So PTMs tend to be low abundant, so you need a lot of cell culture. And therefore, with the TMT, you can really save material, because afterwards you have this combined MS1 and combined intensity. And then if you have like an 11-plex, you need 11 times less cell culture. And the other thing is the missing values per plex are basically you don't have any. So that's also really important for the downstream analysis that we are doing. And also the precision is really high, which is also very important, because many of these PTM changes, they are very subtle, I would say. And to really have not, I mean, if you have two noisy experiments, and you're not really sure if this effect is real or not. And therefore, I really believe TMT is the perfect match for this. Then, as I said already, the PTMs are usually low abundance, so we need to do some sort of enrichment. So some people do IPs, other people for phosphopeptides, like I use like a preparative column where the phosphopeptides will stick on it, and thereby we enrich our peptides that we are interested in. And then, as Tina said, these TMT methods are slow because I also want to do MS3. So we usually fractionate our samples on tips, and thereby we get a deeper PTM coverage. And then we're doing obviously MS3 quantification to really get enough accuracy on our ratios that we want. And I think if you can buy TMT and these mass spectrometers, you should really do MS3. It's just do it. I have no time to go deeper. So the next thing is, then we just put this to MaxQuant. I think is the standard analysis tool for many applications. And then we developed newer technologies to analyze our curves and also visualize it in Proteomics DB. And you will later have some sort of link. You can look at all the PTMs that are available. Okay, so what is dose-resolved proteomics? I teased you a lot. To really understand it, I first want to go though to like how people do it normally. Yeah. So, and what you see here is my favorite example that I often talk about. So it's afatinib in my favorite A431 cell line that's EGFR driven. And what we did here is we basically did standard volcanoplot experiments where we did one single dose. You can always see the single dose up here. And then we had different replicates, and then just look how the volcanoplot basically looks like. So it just shows you phospho sites in that case that were differentially regulated by the drug. And what you see here is nothing is happening. And why is nothing happening? Because we just at a very, very low doses. Yeah. So this is 1 nanomolar. That's not so much. I mean, although some drugs maybe have this. And on the right side, it's a little bit faint, but it will come in better resolution in a moment when stuff is actually happening. But what you can see here, basically, afatinib, it's not hitting to anything. And when we now continue to increase the doses from 10 to 30 to 300 and so on, then you suddenly see changes in the phosphoproteome. And when we now look at target engagements of the drug to certain kinases, then we see EGFR is starting to be hit. And therefore, we see our changes. And what's also interesting is that we know what's the affinity from afatinib to EGFR. And it's in fact 10 nanomolar. So at 10 nanomolar, nothing is happening yet in the cell, but then these effects here, you can really contribute to EGFR signaling. However, if you now continue further, when you go something to 1 micromolar or 10 micromolar, then you have the problem that afatinib starts to hit different protein kinases, and then you get more phosphosites regulated. And if you now would just have this picture, let's say, then you wouldn't really be able to differentiate anymore the signaling that's basically driving this cancer from other kinases. You know, your drug would just interfere. And there's now two really important messages behind this. One is that depending on which dose you use, and if you think about experiments that you may do in your own lab, then you have the problem that you need to decide a priori what concentration you want to pick. And people, unfortunately, usually take something like 10 micromolar or even higher, something 1 micromolar. But in these cases, you already hit all these different off-targets that are available. And the problem is also that polypharmacology is actually a huge problem in these type of experiments. So it's always there. At some concentration, you will have more and more protein kinases that are inhibited by this, and then your picture gets blurred. So this is the reality, and I think also in literature there's many false reported effects just because people use high doses and then misinterpret their own data. But we thought that this actually we can just use to our own advantage, and how we do this is we just appreciate that there are these different affinities to different protein kinases. But then when we now do a series of treatments, so we start with the DSO control, but then a series of different doses, and then look at the different phosphosites that we have, then we actually figure out that when we look at one specific site, that we get something like a really nice dose response curve for each and every phosphosite that's regulated. And what's most importantly is that we have then these EC50 values, that's the key of the entire analysis. And that's basically the concentration where 50% is inhibited almost. It's the inflection point of these two curves, but these are like two concepts people often use, and we like to use the EC50 value. And what's really nice about the curves in general is that we get this EC50 value information basically for free. So we have everything that the volcano plot has. So we have effect direction, so upregulated or downregulated, like how strong the effect is with the effect size, how confident we are, and also how potent we are in the effect. And what we then come, like when you do this now, and now this is the same data set that you had before with afatinib, but this time we actually did this dose-resolved analysis, and then we get this PEC50 dimension. And now what you can see is that we can separate different phosphosites based on the PEC50 value. Yeah, and then when you look very closely, you already see this different distribution that we saw before, like effects that happen very potently around 10 nanomolar, and then effects that happened at higher concentration that are more than 1 micromolar. And we think that because we have this now, we can really nicely separate, and we use this now throughout the paper and show the powerful tools that we can have for this. And I have now a further example for this to really see how in cells this signaling gets transmitted and continues. And for this, we start with gefitinib. It's also an EGFR inhibitor. It's still the same cell line. And when you look at the KinoB profile, so this is the technology that measures the affinity to individual proteins, then we know that this here is something around 300 nanomolar. It's still good, but it's also very, I mean, it's not the most potent inhibitor, but it's still quite selective. And when you now look at the profile of your cells, you actually see that you have a single distribution. And when you now basically look which phosphosites are hidden inside this distribution, you will find phosphosites that were brought there by EGFR, by ERK, and AKT. And that's very important also to understand. Not each phosphosite that's regulated in your experiment is actually a substrate of the kinase that you have. The signaling is just continue to being transmitted inside the cell. So EGFR is upstream of ERK and AKT. But the cell, of course, if you inhibit upstream, then the signal just flows through and goes through inside the cell. But it's really important because it also helps us to deconvolute different signaling pathways. And you can see this in a second. When we now, for instance, go for the next inhibitor, which is afatinib again, you see that we have this very potent EGFR inhibition. But here, the second distribution that we saw here, so this is the EGFR distribution, this is the second distribution, and it's probably related to this P38 kinase family. And if we now look basically on phosphosites here that are known to be a substrate of them, then of course we are already seeing that this effect is here, and then we can basically see that these different effects are happening. And then to make it even more and more complicated, so this is show like a very few off targets. Then we have something like dasatinib, which this is not even all the targets of dasatinib. And there you basically see it's not an EGFR inhibitor in principle. So EGFR is in fact an off-target, but it's like a designated SARC inhibitor. And you see there is some SARC sort of inhibition component inside the cell. But then because it's EGFR driven, the majority is happening here. And then if we can go through our different signaling pathways, you basically can now decode these different things. And then we get these barcodes. And with these barcodes, we basically want to learn how the different phosphosites now can be grouped together, because they should always sort of follow similar trends. And with different inhibitors, we then can basically start to separate them. So that's the basic idea behind dose-resolved proteomics and also Decrypting specifically. And just to show this in principle, so this is a figure from the paper, we use 10 different kinase inhibitors in these cases. There's a few MEK inhibitors, so they are together, and then they have this MEK component obviously in there. And there's different PI3K or mTOR inhibitors. So for instance, dactolizib is this dual inhibitor, and you can see already like every time you see kind of this bimodal distributions, you see that there's multiple targets that are hit. And then there's something what we found in general that's very interesting, in my opinion, is these phosphosites, these numbers tend to be very, very low. So like if we look at the entire phosphoproteome, then it's maybe 1 or 2% of the phosphoproteome that's actually just regulated and doing the job. And this is, I think, quite surprising, especially for like some of these inhibitors that are thought to be kind of dirty drugs, but in fact, on the phosphoproteome in a specific cell line, they're still kind of in this lower number. Except for stavosporin, that's like a really it binds to basically every kinase that's not hidden in 3 seconds. And you see this, it's like I don't know what percentage it is, but probably like 15, 20% of the phosphoproteome is just regulated by this inhibitor alone, and you see this massive distribution that's like all over the place. So good luck to interpret anything coming out of stavosporin treatment. And now that we have these matrix and these different clusters of certain signaling components, we actually wanted to show that it's very powerful and that we can now identify new substrates. So when you look now, this is again kinobate, so we know which targets basically are bound. But then if you look, for instance, for dactolizib and only dactolizib specific responses, we were quite interested, for instance, what is this cluster? And because these are all like the 3 kinases that are sort of thought to be kind of similar, you see that there is a subgroup of kinases which is specific for dactolizib, and they all have this QS motif. And if you now go to our Decrypting data, we actually also see that this SQ motif is kind of exclusively enriched in this pattern. And if you now go for these different sites, you find, of course, then very known sites like this BRCA1 site. It's very famous with this also famous SQ motif. But then there's also many other sites, like for this protein that has obviously the same motif and quite similar potencies. And then therefore you can infer that this one here is probably being phosphorylated by either of these 3 kinases. And that is basically, we can do this for the entire proteome and basically look at different phosphorylation events. That's one thing. And then we can basically also integrate all the data together. It's a little bit messy. I will only partially go into detail. So what you see here is basically different pathways that can be engaged by different drugs. The drugs are in this grayish bubbles. And then when we now look for one specific inhibitor, for instance, you can now basically understand the mode of action of this. So this is dactolizib. You see this very strong mTOR component. And what I found, for instance, very interesting is that sort of you treat dactolizib, but then the cell at higher concentration has this counteracting effect. So what you see here basically is that this is upregulated MAP kinase signaling, while mTOR most of the time is downregulated, but there's also some components of mTOR which is upregulated. So if you think most of the cancer drugs are well understood, they're really not understood. So there's like all over the place. While, for instance, AKT is going down and actually quite potently. So that's what it's supposed to be, but there's way more to explore. Just to say, it's not only working for phosphorylation, it's working for other PTMs. So we have acetylation. And here, for instance, it's also really nice, like big pharma should really also tackle this approach. And what you see, for instance, is these two different inhibitors, and you see this different methyl urea groups. And just by changing the position from here to here, you change the affinity to this acetyltransferase. And you see this also when you now look at the phosphoproteome. What I like actually is that it's a huge effect. It's like 15% of the acetylome is changed just by this very potent one acetyltransferase. Then, just to kind of give you the last idea of what is possible with the technology, and this is two-dimensional Decrypting. So what's the second dimension? It's time. So what we started to do is not only basically getting this dose responses, but actually we look now at dose responses over time. And that's, for instance, quite interesting for this bortezomib inhibitor. That's a proteasome inhibitor, and it's covalently binding. So we actually also start seeing sort of shifts in PEC50 values. So that's all can also be now like a different dimension again that you use to separate effects, because if you inhibit something and then you see these PEC50s moving, and then some PEC50s also moving or not moving, you can further start to separate things. But of course, it's a lot of more work to then not only doing a lot of treatments, but also doing a lot of treatment times. And what also biology-wise was quite interesting is that if you now look at this proteasome inhibitors, then actually on the full, I mean, this is phosphoproteome, but first I tell you the full proteome. So the full proteome is really not changing for the majority of the treatment time. So only if the cells are really dead or almost dying, then like huge full proteome changes are happening. But when you look at the phosphoproteome, so this is what you see here, then you actually see already in a very short time the phosphoproteome response to this proteasome stress. In particular, if you look at ATF4, for instance, that's the key transcription factor that sort of starts the proteasome stress when you have lots of unfolded proteins and the cell tries to rescue this. But of course, it's a hard job if your proteasomes are all inhibited. And we also could see that why did the proteins basically not change over time is that quite early here, like the majority of this upregulation is actually coming from phosphosites and proteins that are important for protein biosynthesis. So that was also for us quite interesting to see that the cell feels like, okay, protein degradation is not working. So we should first try to solve the proteins that are there, and then they really stop the protein biosynthesis. With that, I'm basically done. So I just want to summarize quickly what we've seen. So the first thing is Decrypting profiles are more informative. I hope you all agree to this. So we get this extra dimension and you basically always get it for free. So when you do your own experiment and you can use this extra dimension, you should use it. Then it also, that's something I didn't mention yet, but it's also true that we get really nice profiles for not regulated data. And we have then this high quality negative data set. And that's also something that's very useful for all sorts of machine learning tools that are around. And people usually just report what's regulated, but it's also very interesting what's not regulated. It's scalable. So we can now do it very fast. We have many robots now also at the chair that are supporting us in many steps. It's very reproducible. We can now also learn really something about the cellular mode of action. And we have, you can really use it for two-dimensional things and for this structure-affinity relationship experiments. And apparently the reviewers also liked it. So it was quite some people who worked on this project. So it started in 2016 with Ben and some students. Then Jana took over, and Svenja also did lots of these experiments, and then I kind of pushed it to the end. And so it's really like a huge group effort, and that's why you see many people on the paper. And with that, I'm done.